Hi my friends, welcome to my channel that is ACE Biology. Now today we are going to discuss about the structure of DNA and RNA. Now we know that DNA is deoxyribonucleic acid and RNA is ribonucleic acid. So the basic structure of both the DNA as well as the RNA molecule is somewhat similar because both will consist of the pentose sugar, the phosphate group and the nitrogenous base. Here in the diagram also you can see that both the RNA nucleotide and the DNA nucleotide consists of this phosphate group, the pentose sugar group and the nitrogenous base. Here also the same groups are present. But as the name suggests, the difference lies in the pentose sugar group. Here you can see that at the 2' position of these pentose sugar group, in case of DNA, you are having only a hydrogen atom. But in case of RNA, you are having a hydroxyl atom. That is, the OH group is present here. So this is the difference between RNA and DNA. Now another difference between these will be in the nitrogenous base. Here you can see that in case of RNA nucleotide we are having adenine, guanine, cytosine and uracil. And in case of DNA nucleotide it is adenine, guanine, thymine and cytosine. So here you can see that in case of RNA, thymine will be replaced by uracil. Now coming to the structures of the nitrogenous bases that are present in DNA and RNA. Now here you can see we are having adenine, guanine, thymine, cytosine and uracil. Now adenine and guanine, these will come under purine. These are purines. And thymine and cytosine as well as uracil will come under pyrimidine. Okay. Now, in our in RNA molecule, this thymine group will be replaced by uracil. So these are the two differences existing between a DNA molecule and an RNA molecule. Now we come to the question like why is this difference present in case of RNA and DNA? Now we know that the DNA is a genetic material. It will store all the information that is to be passed from one generation to another generation. So we can say that it needs to be stable. Now what happens in our body? There are a certain repair mechanisms that are going on. These repair mechanisms are the role of these repair mechanism is to see whether all the DNA replication that is occurring in our body is correct or not. So what happens in these repair mechanisms? There will be certain enzymes. So by chance in some cases if the cytosine gets deaminated, then it will convert into uracil. Now this repair enzyme that is functional to check whether the DNA replication is being carried out in a proper manner or not, it won't be able to discriminate between the original uracil that is already present and the uracil that was formed due to the deamination of cytosine. So that's why this uracil was replaced by thymine. Somewhere it makes the DNA to be more stable as compared to the RNA. Like even if some error occurs, this RNA, these repair enzymes will quickly catch up the error and they'll rectify it. Now the other question is why RNA was replaced by DNA as a genetic material. Here the answer lies in the difference itself. Now we know that the DNA is a genetic material. Like in case of some viruses, you will find that RNA also acts as a genetic material. But we know that RNA is highly stable, and in viruses also, these RNA, they are more prone to mutations. They'll change a lot. But for us, the DNA, we are having DNA as a genetic material. So DNA is quite stable. So what is that factor that makes it stable is this 2' OH. Like we had already discussed, the difference between the DNA and RNA is the 2' OH which is present in the pentose sugar group. So what happens is at this 2' position, in case of ribose, will be having the OH group, and in case of DNA, it's only the hydrogen atom which is present here. So this OH group is very reactive, and in case of any alkaline medium, it has the tendency to form cyclic structures with the alkali metals. So what will happen? It will inhibit the replication, thus making this RNA very unstable. That's why we prefer to have DNA as our genetic material. Now, coming to the bonds that are present in a DNA, the two important bonds will be the phosphoester bond and the glycosidic linkage. Now, as already discussed, the DNA will consist of the nitrogenous group, the pentose sugar, and the phosphate group. This phosphate group is attached to the pentose sugar by phosphoester bond, and the nitrogenous base is attached to the pentose sugar by glycosidic linkage. Now, apart from this, we are also having some other bonds like hydrogen bonds, which is present in the DNA double helix that we are going to discuss further. Because we know that the DNA will exist in the double helical structure as said by Watson and Crick. Now, the double helical structure of the DNA will consist of two strands. These two strands will be complementary to each other. Like here you can see one strand is 3' to 5', and the other will be 5' to 3' here. So these two strands are complementary to each other. Now here we are having the adenine, guanine, thymine and cytosine. Now adenine will always pair to thymine and cytosine will always pair with guanine. Now these pairing is like the bond present between adenine and adenine, thymine and guanine and cytosine will be hydrogen bonds. Between adenine and guanine we are having two hydrogen bonds, and between thymine and cytosine we will be having 3 hydrogen bonds. So it is clear that the bond present between cytosine and guanine will be stronger as compared to adenine and thymine. Now there are other parameters that are quite important in DNA double helix, like the diameter of the DNA. It is around 2 nanometer. The distance between two adjacent nucleotides present in a DNA is 0.34 nanometer, and the pith of the DNA is 3.4 nanometer. Now what exactly is the pith of the DNA? It is just the height per complete turn of the helix. Now here you can see this is a single turn. So the height of this turn will be 3.4 nanometer. This diagram represents pDNA because it is right-handed. So here we can see, here it is going like this and like this. So here you can see that there is a clockwise rotation. So whenever there will be a clockwise rotation, it is right-handed. It will be a right-handed turn. Now apart from this, we also get to know these two terms which are generally used in this helical structure, that is the minor groove and the major groove. Now these are the consequences of different geometry of the base pairs. What happens? The glycosidic bonds of the base base pairs are not diametrically opposite to each other, due to which we are having these grooves. Now the minor groove will be a little narrower as compared to the major groove. Because of this glycosidic bonds, we came to know that in our DNA there will be formation of some of the grooves which will be either minor or major. Now apart from this, some other conformations also conformations also exist due to the orientation of this glycosidic bonds. Like here, in case of DNA, we'll see that there will be two kind of conformations, the syn conformation and the anti-conformation. So this difference arises only due to the presence of a particular atom just above the pentose sugar group. Now here you can see in case of syn conformation, we are having this nitrogen atom which is present just above the pentose sugar group. Now this is N3, the third nitrogen. Okay. And here in this case, now here both of these represent purine nitrogenous bases. Okay. So here in this case, like in anti-conformation, we'll be having a hydrogen atom here, and it is the 8th hydrogen atom. At the position 8, this hydrogen atom will be present, and it is just above this pentose sugar group. So whenever we are having hydrogen atom just above the pentose sugar group, it will be anti-conformation, and whenever we are having a nitrogen atom group, that is N3, we'll be having syn conformation. Now we are in nature, we are having different forms of DNA. The most important are the B form, A, and the Z form. There will be a little difference between all these forms in the base present per helical turn, the grooves, and the helical sense, like whether it is a right-handed turn, whether it is having a right-handed turn or a left-handed turn. So in case of B DNA and A DNA, the helical sense will be right-handed, that is the clockwise, and in case of Z DNA, we'll be having left-handed helical turn. Major groups are present in both bDNA and aDNA. It will be absent in zDNA. Also, the bases that are present in a helical turn will be 10.5 in case of bDNA. In case of aDNA, it is 11, and in case of zDNA, it's 12. And the helix rise per turn, it is 3.4 angstrom in case of bDNA, 2.6 in A, and 3.7 in zDNA. Also, the syn and anti conformations that we had already discussed. So what happens in case of B and A DNA, anti is the preferred conformation. In case of Z is a DNA, we will be having anti for pyrimidines and syn for purines. Now, what happens under normal physiological conditions, this bDNA exists, but if there will be any kind of change in the environmental condition, it is seen that these bDNA gets converted into aDNA or zDNA. For example, in case of dehydration, this bDNA will get converted into A form. Now, it does this just to protect it from the external environmental conditions. And also, whenever we observe any high salt concentration, it is seen that this B form will get converted into the zDNA. So that's all for this DNA structures and the RNA structures. In our next video, we are going to discuss about DNA topology. So if you like my video, please hit the like button and don't forget to share and subscribe. Thank you.